Hello, this is the first part of Module 2, and we're talking about taxonomy today. So what is taxonomy, why does it matter, and more importantly, why does it matter to you? This is the outline, and just like last time, you can click on any of those segments over there and jump to that part of the video. And today we're going to be talking about what is taxonomy, what is monophyly, paraphyly, and polyphyly, and why are paraphyly and polyphyly problems. We're going to be talking about how you actually determine relationships between organisms, and then talk about why does all of this matter. So, what is taxonomy? In Greek, taxonomy just means arrangement method, and if you really want a nice scientific definition, it is an academic discipline of defining groups of biological organisms on the basis of shared characteristics and giving names to those groups. So basically, you take a whole bunch of things and you figure out how they're related to each other, and then you give names to those groups, and then as you get further down and more specific, then you actually end up giving names to species. And it really just gives order to the whole naming system that we have for all organisms on the planet. Here is a pretty typical example of a taxonomic classification of an organism. So we're going to start with the honeybee, or Apis mellifera, and it was coined, its scientific name, by Linnaeus in 1758. Most of the times when you have a scientific name, you'll have whoever named it and the year that it was named. And we'll get into this a little bit later, but sometimes you get multiple names because multiple people found the same thing and didn't realize it had already been named, and then people have to go through and pick which one came first, and then there's a whole process. But every time you see a scientific name, there should be someone who is associated with that scientific name and the year that they had named it. The honeybee is in the kingdom Animalia, like you, me, whales, fish, salamanders. It's then in the phylum Arthropoda, like crustaceans like your crabs and your lobsters, other insects, things like pill bugs and scorpions. Then it's in the class Insecta, which includes mantises and grasshoppers and anything with 6 legs. Then it's in the order Hymenoptera, which is a group of bees, wasps, and ants. Then it's in the family Apidae, which doesn't have a really good common name. It's just kind of like your bees. And then it's in the genus Apis, and then the species Apis mellifera. Species is always the genus name and the specific epithet together. And it is always italicized. Genus and species are always italicized. All of the other classifications are capitalized when you write them out. Sometimes you want to talk about an organism and the specific grouping is too specific and the broad grouping is just too broad. And so we have supers and subs, which are little groupings in between the major ones to help broaden and or narrow what you're talking about. So I've picked a hemipteran, this is a bug, it's a leafhopper, as an example. I'm going to be going through the classification of this leafhopper in the same way that I did with the honeybee. This leafhopper doesn't have a common name. So, and leafhopper is kind of a general term that's given to a lot of different groupings, and so, sorry, the common names aren't going to be that helpful, but this is part of the reason why we have taxonomy. So, this leafhopper is in the kingdom Animalia, the phylum Arthropoda, the class Insecta, the order Hemiptera, and Hemiptera includes cicadas, leafhoppers, and your true bugs. It's in the suborder, and a suborder is a classification directly below order, and it's in the classification Auchinarynca, which is just talking about where its mouth parts come from, but also in this group are cicadas and leafhoppers. It's in the superfamily, which is a classification directly above family and directly below suborder, and superfamily, and its superfamily is Membrastoidea. And Membrastoidea includes things like your tree hoppers and leaf hoppers. Then it's in the family Cicadelidae, which are leaf hoppers. Sometimes they're also called sharpshooters. And then it's in the subfamily Cicadelinae, which has no common name and just includes these leaf hoppers that look very similar to this. And then you have its genus and species, and it has no common name. But this would be the classification introducing suborders, superfamilies, and subfamilies. In some cases, there are also superorders that we talk about, but it's not as common. Here's a quick cheat sheet to your endings in the taxonomic grouping, so you know generally what kind of grouping you're talking about if you see a name or you hear a name. Please note that this only applies to animals because animal people and the plant people and the fungus people and the bacteria people, none of them talked. And so everyone has their own different system based on what larger grouping you're talking about. And it's kind of unfortunate, but that's the way it is. So orders, and only with insects, and only most of the time, ends in terra. And terra just means wing, and a good example is Hemiptera. There are some orders like Odonata, which are your dragonflies, which don't end in terra. And there are some suborders that do end in terra. But if you see terra, you know you're talking about a generally larger classification. The superfamily ends in oidea, O-I-D-E-A, always, forever, through all taxonomic groupings in animals. And an example is Membrasoidea. Family endings are ide, I-D-A-E, and an example is Cicadelidae. And subfamilies always end in ine, I-N-A-E, and an example is Cicadelinae. With your insect identification, you're really only expected to identify down to family. And that's because while family is a standardized taxonomic ranking, families are not created equal between insects and lizards and, say, fish. And I think that's because just that insects are far, far more diverse, and within one family, most of those insects aren't particularly distinguishable, especially if you get down to genus. They're species that are almost indistinguishable from each other. And most of the time you do that by dissecting genitalia. And identifying down to species doesn't really tell you any more information about the insect than it would just down to family. And that's different than, say, birds or mammals, where you have like specific guides for, and you can identify them down to species because they look really different and they're not as diverse, and so you have bigger differences in your species. So when you do your identification, you're only expected to go down to family, and if you try and ID down to species, it will most likely lead you astray. So just use the characteristics to identify down to family. That's what we do with insects, and then you'll be good to go. Okay, and now we're going to talk about larger groupings and the kind of the ideals behind taxonomy. And we're going to do this with three major groupings: monophyly, paraphyly, and polyphyly. The first way to group organisms is with a monophyletic group, and a monophyletic group is a group of organisms which consists of a species or of a grouping and all of its descendants. And this is what we strive for in taxonomy. This is why things move around sometimes and things change names. It's because as better and better techniques come out for identifying and characterizing organisms, we try and move them around to get these monophyletic groups, so it shows a clear evolutionary path from this organism to that organism to this organism to these like 8. And so, for instance, if we talk about a monophyletic group, I could talk about insects, and we all know that insects have 6 legs. And then I could talk about beetles, and everyone should know that beetles have a hardened shell called an elytra. And so when I say these names, because they're monophyletic groups, you can get very clear images in your head about what these organisms should look like. Sometimes things go horribly wrong, though. Just because a lot of this taxonomy work has been done in the past, wherein DNA wasn't a thing that you could easily and cheaply study, and so we were based on things like morphological characteristics. And just like people could go easily astray with stick grasshoppers looking like walking sticks, some characteristics look really similar, and so one grouping would be tossed into another group or taken out of a group based on convergent or divergent evolution. And one example of this going wrong is a paraphyletic group. A paraphyletic group is a group of organisms consisting of all the descendants of the last common ancestor except for a small number of monophyletic groups of descendants. And what does this mean? Basically, this means that you take a group of organisms and you lump them together because it's convenient and they share pretty similar characteristics, and you conveniently ignore the ones that don't look quite right. And a really, really great example of this are reptiles. So if I talk about reptiles, you're like, oh yeah, crocodiles, snakes, lizards, legless lizards, alligators, monitor lizards. And it turns out that birds are actually reptiles, because birds are the most recent descendant from the ancestor that had the amniotic egg. Mammals are also an ancestor from the amniotic egg-bearing creature. And so, by defining the group reptiles, you have left out mammals and you have left out birds because it's convenient because we aren't cold-blooded and scaly and look lizardy. The next way a thing can go wrong is a polyphyletic group. And a polyphyletic group is a group of organisms consisting of those which have been grouped based on character state which have converged or reverted as to appear to be the same but which have not been inherited from a common ancestor. And this basically means that you take two different groups and you lump them together because they look basically the same and you have no idea about their evolutionary history, but you think they should be together because they have the same characteristics. A really, really extreme example of this would be if you took mammals and birds and lumped them in the same group because we're both warm-blooded. What actually happens is this happens a lot with insects because you have a lot of convergent evolution, so you'll get similar features because the environment, whatever the insect was in, has pushed and put selective pressures on a certain kind of body form. And another really great example is if you have cave fish. A lot of cave fish lose their pigment and they lose their eyes just because neither are really needed in a cave environment and it's really easy to lose them. And so people will lump them together because they're like, oh, they both have no pigment and they both have no eyes, they must be related, even though evolutionarily they came from two really distinct lineages. This can also happen if you find a legless lizard and accidentally call it a snake. A legless lizard is not technically a snake. It is its own grouping, but because they both are long and skinny, have no legs, and you might be likely to put them together. And polyphyletic grouping is another way that you can accidentally go astray from your perfect monophyletic vision. So how do we get these perfect monophyletic groups? We use these things called synapomorphies. And a synapomorphy is a trait that is shared by two or more taxa and their most recent common ancestor whose own ancestor does not possess that trait. So, for example, in fact, the only real way to explain this in common terms is to use an example. So, for example, beetles. You think of a beetle, you automatically think of like a ladybug or a firefly or something with the big hardened shell over their hind wings. And that hardened shell is called an elytra, and that elytra is the synapomorphy that separates beetles from all other insects. And so when I say beetle, you should automatically know that you have this group of organisms that even though the elytra may be reduced, you have some sort of armored and protected hindwing. A couple other examples of synapomorphies are just having 6 legs. So your hexapods are separated from things like your millipedes, for example, because they have 6 legs. Not all hexapods are insects, but all insects are hexapods. And so what separates insects from hexapods are that insects have the Johnston's organ in their antennae, which picks up mechanosensory cues from around them. Pterygoda, or winged insects, are their own group because they got wings. And then you have a group called Neoptera, which are the new wings, and that's because they gained a muscle in their wing that allowed them to fold their wings flat over their back instead of having to hold them straight out or up like dragonflies or damselflies. And so each grouping, starting with the kingdom all the way down to species, each group has a synapomorphy to separate out that point or that common ancestor and all its descendants from everything that came previously. A lot of these synapomorphies in the past have been based on morphological data. And this is why things keep changing and moving around, because since DNA and DNA barcoding and DNA sequencing has become a lot cheaper, we're using that in addition to morphological data to try and move some of these things around and get a little bit more stability in our taxonomic tree. While genetics are super helpful right now, they aren't going to replace morphological data anytime soon for two main reasons, really. The first reason is that it's too expensive. While genetic decoding and stuff is getting a lot cheaper, by no means do we have the resources to catalog genetically like 300,000 beetles. Like, we have 300,000 beetle species named and in museums and stuff. There's no way that we could actually go through and with the funds that are given to us from the government go and actually establish genetically where they belong. So the money isn't there right now is the first main reason. And so the money is being reserved for things that are economically important. Another reason why genetics aren't going to take over anytime soon is because they aren't as specific as we think they should be for species delineation. And this is especially true for really closely related groups, like especially species in the same genus. And a really great example of this is in caliphorids or blowflies, which are really, really important for forensic entomology. Because basically what happens is you have a dead thing out in the forest and then a blowfly finds it and lays its eggs on it, and then by the time you find the body, you probably find pupa or maggots on it. And so you collect your maggots and your pupa, you rear them out to adults so you can identify them. And it's really important that you identify them down to species, because then you use all that species-specific development information to determine how long your body has been out in the forest. And so usually when you figure out what species your fly is, you call in an expert or you sit through the keys yourself, and the keys aren't exactly the easiest thing to use in the world, especially when you get down to species, because it's like, what color are these random mouth parts, and how many bristles are on this obscure structure, and if you have a male, what do its genitalia look like? And it's not really something the average Joe Smo is going to be able to do and get through the terminology particularly well. And so people were looking for something to be able to use instead of these morphological characteristics. And so one of the groups of genes that people used were the cytochrome oxidases. And this was really great in concept, although it turns out that for certain groups of caliphorids, it doesn't work well at all. So, for example, some Califora species, it works fine, and they're all pretty much the same, but they're different enough to be able to tell different species. But groups like the Lucilia, which is a different genus of blowfly, they're all the same, or most of them are the same, and the only way you can tell the different fly species apart is basically where you got them, because you'll have a neotropic species and a North American species that have exactly the same gene in that spot, and the only way that you can really tell the difference besides morphological data is that you picked it up in Ecuador or you picked it up in Connecticut. And so we hope that these specific markers are actually species specific, but they aren't always as specific as they'd hope they'd be, so you have to rely on other data like location data and morphological data. Despite the problems with genetics, it's actually been really, really helpful. So one of the ideas for a while was that insects were most closely related to millipedes and centipedes, and then they lost legs, and legs turned into wings, and legs turned into mouth parts, and they lost them. But now the idea with genetics coming up is that insects are actually just crustaceans that have dominated the land, and so insects are actually more closely related to pillbugs and lobsters and crabs than they are centipedes or millipedes. And that's where genetics is going right now, and that's called the pan-crustacea theory. Another place where it's actually helping a lot is kind of these bigger groupings that we thought we had, or organisms that we thought were in these bigger groupings that now aren't so much anymore. So for example, if you've had taxonomy in the past like 3 years, and you've probably learned that termites were their own order, or they were in their order Isoptera. Well, it turns out with genetic testing that Isoptera doesn't exist, because termites are actually just eusocial cockroaches. And termites were originally put into their own group because they look different, they have a really different behavior than normal cockroaches, but then with all these genetics, it's showing that these termites are showing up in the middle of these cockroach phylogenetic trees. And so fairly recently, termites, like in the past like 2 or 3 years, it's been accepted that termites are now cockroaches. As to their exact official status, people are still arguing. Right now, UGA is teaching it as a superfamily, Termitoidea. Although exactly, again, depending on where you look on the internet, people have different ideas about what it should be. The real question is, why does this all matter? Why do we care if we have monophyletic groups, and why do we care if it's all organized, and why do we care about naming things? So one of the goals of taxonomy is to provide consistency. While the plant people and the animal people and the bacteria people haven't really talked to each other very much, and so you can have overlapping scientific names between animals and plants, within the animal community, one scientific name is only applied to one animal, and each animal only has one scientific name. That is one of the other goals of taxonomy. Sometimes it's effective and it works, and sometimes it doesn't so much. So, for example, we have the eastern red bat, and it was originally in one genus and then got moved to another genus, and so its scientific name changed. And so a lot of people complain about taxonomy all the time. So they're like, we learned this one name, but then it got moved, and now it's a different name. And that's kind of annoying, but again, it goes to that goal of trying to make everything a monophyletic group. Sometimes you'll get things called synonyms. It happens a lot in insects because so many people are going out and trying to identify insects and no one has time to like double check all of them. So sometimes one animal will get several different scientific names because different people found it and didn't realize it was named by someone else already. But there's a whole committee that their job is to go and find those, what they're called synonyms, figure out which one was the first one, and if it was accurately placed in the right place, and then assign that as the official name. So on things like the Tree of Life Project, or if you look up scientific names on Google, a lot of times websites will come up that are sponsored by the government and say what the scientific name is and all the synonyms it's had, or all the other different scientific names it's been called, and which one is valid. Another goal of taxonomy is to remove common names, because common names are awful unless you're a bird person. Apparently all the bird people in the world like got together and like, it'd be really perfect if we actually had only one common name per bird and everyone called this bird the same thing. And they did, and that's great because you don't need to learn the scientific names of birds because the common names are actually worldwide recognized, and that's awesome. Unfortunately, we aren't so lucky on anything else. So, for example, insects from up north where I'm from, you have like the red bug, which is a pest on apples. And then down here, I'd heard a red bug, and apparently it's a chigger. And so red bug applied to two totally different groups, not even different species, like different huge groupings. One is an insect and one is not even an insect. And so this happens a lot, especially in insects, but in a whole bunch of other groups too. And so another benefit of taxonomy is to give people a name that scientists can use all together, and that will only refer to that one organism. Recently, people have been dabbling in genetic barcoding, where you take an organism or a species and then you tag a part of its genome that is very, 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 very species specific, and you put a barcode on it and you upload it to GenBank or some other national database. And with that DNA barcoding, you have attached to it every scientific name or every common name that that organism has had forever. And so if you find an organism and you don't know what it is, you can sequence its genome, put it up, and it'll match to this known sequence in this known database, and you'll know exactly what it was and its naming history forever. And so that's really awesome. And so that's what we're working on, and by we I mean taxonomists are working on now. Another benefit of taxonomy is that it's providing an evolutionary roadmap, and so we can see how things have evolved over the past X billion years. So one of the really cool examples of this is bird evolution. As we're digging up more fossils and putting them into places based on their characteristics, we can more easily see how these major morphological features have evolved. And this is why it's really important when we find missing links, because it fills in our fossil record and makes our taxonomy a lot stronger. Another great thing about taxonomy is because you're grouping things together based on characteristics and synapomorphies, it tells you something about that group. So if you find an organism in that group but you don't know anything about that organism itself, you at least know something about it based on the group that it's in. For example, if you don't know what a cuttlefish is, if I tell you it's a cephalopod, then you know it's related to octopi, has tentacles, and is pretty smart and most likely can change colors. If, for example, I gave you an echidna and you had no idea what that was, but I told you it was a monotreme, and you knew that platypuses were monotremes, and you realized it was an egg-laying mammal, and because it was a mammal, it had mammary glands, and had fur, and all of these other basic characteristics that you associate with that larger grouping that it's in. And that's, I think, is one of the strongest points of taxonomy. So you can find an insect and be like, oh, this is a praying mantis, and already know things about its biology just because you've placed it into the group of mantises, even if you don't know anything about that particular mantis. The last thing, and what's probably most useful for you, is that you can identify unknown organisms because you have this taxonomic system already set up where you go through a key or an identification book or a field guide, and there are specific characters and synapomorphies you look at your creature to be able to place it into a group. And once you place it into a group, then you know a little bit about its biology. So, for example, we have this beautiful, majestic creature here, which I won't reveal what it is. If you've had an entomology course before, I'm sure you could probably identify it pretty quickly. If you haven't, then you might think of it the way I first thought of it when I first saw it, and was kind of like, wow, this thing looks like a centaur. It's pretty much like the head of a mantis and the coloration of a wasp, and kind of with a fly-like body, and it's really confusing. But as we'll see when we get into our insect identification video, this is actually a pretty charismatic and easily identifiable organism, and I think you'll be surprised where it ends up. I hope you learned a lot from this taxonomy video, and that it helped clarify what taxonomy is and why we use it and why it's important. If you have any questions, feel free to contact me. This is obviously a lot more complicated than the simplified version that I have here. I'll do my best to answer you, but right now a lot of taxonomy is philosophy, which organization methods are the best, and why, and who thinks what and why. But if you find any of that interesting, I have some resources for you, and you can contact me and I can get them to you. So overall, I hope this was really helpful. I hope that you're excited to find out what this mystery creature is if you don't know it already. And I hope you're excited because the next installments are going to be identifying different non-insect arthropods and then identifying different insects.